Okay, introduce Karan.
Calgary.
Okay, introduce Karan.
Guess we're live, are we?
Hi, Jacinta.
Hi.
Hi, ma'am.
Hi. Soma, how are you?
I'm good. You're beautiful.
Oh, thank you. You're very kind. So, Soma, where are you based?
I am taking a break. I'm thinking about getting into PhD. Shubha has not been able to convince me till now, but maybe I will. I did my master's and I'm taking a break
So, what did you do your master's on?
in biotechnology.
Biotech.
And I also went ahead and did MTech in biotechnology.
Wow.
And
You do
so,
need a break.
yeah. Actually, I like teaching. So for some years, I was teaching. I had a tutorial kind of. It's called. It has a website, the iqeducation.in. You can find it there. We were teaching grad students, and now it's closed because of the all the pandemic and all.
COVID stuff, yeah.
Yeah, so maybe online, maybe we can teach online, and let's see how it goes. As of now, I'm totally focused in building bioingene.
Great. No, you guys are doing a great job. I mean, this is massive number of registrations, and you're able to pull these people. I think this is a great way to look on the positive sides of the pandemic. You know, it has
Yeah.
connected the world in different ways we've never imagined before. So
Yes, definitely. So many
and thanks
things are
to you guys for
changing.
thinking of all these ideas and doing the hard work. So
Yeah, we are very lucky that our friends have, out of the goodness of their heart, they have joined us because it's totally non-profit and no money involved whatsoever. So very, very glad that our friends, we are all together in this and doing this. And since it's free, webinars are free, so we encourage. I mean, more students are being encouraged. They don't have to pay for. They don't even have to think twice before registering.
that's how
So
education should be. You know, education
yeah.
should not be like there shouldn't be a price tag on, you know, education. This is one of the basic things I think humankind can easily afford, but probably we've made this the biggest business at this moment.
That's so true.
It's really sad, but hopefully people like you will change the world, and
We
this
are,
is a real impact you're making.
and
No,
thank
I'm
you
very
also.
serious about it, to be honest. Like this is a real impact. You're doing a very big thing. You don't probably realize it, but yes, it's amazing.
And we are also very thankful to all the scientists who have come here as a speaker, and even they have encouraged us, and we are really, really thankful. I mean, we are nothing. We were in the beginning. I mean, even now we are actually nothing. So still they trusted in us, and okay, let's these are new, young, I mean, we don't have a reputation as a big website or anything. We're just very, very new to this. So we are very lucky and we are very fortunate and very, very grateful to all of you. I mean, the speakers, the renowned scientists. I mean, thank you so much for
No,
trusting us.
it's a pleasure. Thank you. So, Shiva, I see that we are already live on YouTube.
Yes. We're live, but we are live, but we are waiting for 5 minutes because it's started at 11 a.m. And I also add you in LinkedIn. Your name is inserted on our invitation, or you can check it.
Thank you. No, I will share it as well.
No,
just in
yes,
there's
for such purpose.
And also, so you want to check to share the slides or something like?
You can try, but I know you you habituated. I know.
Okay, so just want to make sure that everything works, you know, because I've got a couple of screens in here, so
Yeah, okay, no problem. Take it.
Oh my God. It's gonna be a bit of a problem for me because it's taking my, wait a minute. I've got a few screens here, so that sometimes it gets a bit confusing. Okay, seems like I'll have to move, I'll have to just move to one computer. Just give me 1 second. I'll
No
just switch
problem.
off my,
No problem.
because otherwise it's going all over the place and I don't want that to happen. Just gonna close everything. Okay, it's still going on to one. All right, now we are just with my laptop. Okay. Okay, so Shivo, let's
Yes.
try and share screen
Okay, ma'am. Okay.
and
fine.
see if my laptop thing is working.
Okay, fine.
Looks good.
It's good?
Yes.
Awesome. So I can just see you guys as participants for now. So is there anything else I'm going to see on the screen once you?
You need, ultimately, we collect the questions from YouTube and paste it in the chat box during the question answer round. Actually, we collect them and search which one the appropriate for you. And then only selected few we can paste here, but you have the freedom to answer any of your choice.
Okay, sounds good. And you guys want me to keep my video on, of course, during the seminar?
Yes, it will
Yep.
be better.
Okay, super. Do you want to keep the screen share on now or shall I stop it for now?
If initially you please stop it because we need your face
Yep.
first, then screen.
Yeah, no, all good.
Give me just 1 minute. I send all the attendees to your live link by mail.
Sure.
Jacinta, is everything is okay?
Yep.
Okay.
Do you want me to stop?
Just 1 minute. We already reached about 400 people in YouTube. So now we can start. So, okay, I'm going to start now. Huh. Liberty, are you there? I think, yes. Okay,
Yes, I'm here.
okay, Liberty. Okay, ma'am, we are going to start. Hello, everyone. Welcome to our Bioingene live webinar series. Today, we have our speaker, Dr. Parwinder Kaur from Australia. Jacinta will introduce her. Soma will interview her. Liberty and Neha will collect all of your questions from YouTube chat box, and we make sure that your questions are collected from there. So please hold your question. If your question is relevant to our today's talk, please paste it and we collect from our YouTube. We have some announcement because now we arrange a soft skill webinar because we all thought that soft skill is a very important nowadays for the academia and for our education system, and most of the developing education system not have such strong soft skill facilities. So we try to include soft skill webinar, though it is not related to plants, but many of the viewers are requested for this type of webinar. So the registration has begun from yesterday night. So you can register for the soft skill webinar with other webinar also. Jacinta, please introduce our speaker.
Hello. Welcome to today's webinar organized by Bioingene. Bioingene is a non-profit organization created to promote plant research worldwide. The webinar series has been designed to build a platform from where plant scientists can present their research to the world. We hope future scientists can gain perspective and inspiration by listening to the esteemed plant researchers talk about their scientific accomplishments and their thoughts on the future of plant sciences. We are grateful to the many renowned scientists that have accepted our invitation to share their research insights with us. We have received a huge response of more than 3,600 registrations for today's webinar. We welcome the audience and thank them for their interest in Bioingene. It is our request to all viewers to please hold all questions for the question and answer round at the end of the presentation. You can type your questions related to today's talk in the YouTube comment box. You can apply for a certificate of participation via the feedback link, and the password will be provided after the presentation. Submission of the feedback form is mandatory to receive a certificate. The topic for today's webinar is Role of Agricultural and Environmental Biotechnology for a Sustainable Future by Dr. Parwinder Kaur, who is an associate professor and the director of DNA Zoo Australia at the Faculty of Sciences in the University of Western Australia. Dr. Parwinder Kaur leads an innovative translational genomics research program that aims to translate fundamental science into ready-to-use solutions across the agricultural and medical sectors. Her DNA lab team enables research to span the spectrum of scientific activities beyond the traditional lab-to-landscape model, using new-age technologies such as CRISPR, single-cell, and 3D genomics. With DNA Zoo Australia, she is on a mission to provide genomic empowerment to unique Australian biodiversity, facilitating conservation efforts for the threatened and endangered species. She has made substantial contributions to the field of biotechnology and was appointed as UWA's Be Inspired for Agricultural and Environmental Biotechnology in 2019. Her studies tracking genomic variation to breed low methanogenic forages in Australia provide a new paradigm for reducing the environmental footprint of ruminants. She has been honoured by the prestigious Science and Innovation Award for Young People in Agriculture, Fisheries and Forestries by the Australian Academy of Sciences in 2013. A joint venture between UWA and the world's biggest pasture seed company, PGG Rights and Seeds, resulted in demonstrating the impact at the paddock level. Also, DNA Zoo's innovative work developments won the Microsoft AIs for Earth Award for 2019 to 2020. We are happy to have her with us today, and I now request Dr. Kaur to begin your presentation. If you could kindly share your screen with us. Thank you.
Sure. Thank you so much. Before I get started, to Bioingene and Shuvo and Jacinta and the entire team, thank you so much for organizing. This is a massive effort, and all the listeners and who've joined us from all over the world, I would like to say thank you so much for giving your time, and let's get started without wasting another moment. Is everybody able to see my screen okay before I get started? Shuvo,
Yes,
can you please confirm?
Yes, ma'am. It's okay. Perfect.
Okay. Super. So today I would like to take you guys on a little journey here with me. And before we get started on that journey, I would like to share with you guys the location where we are based. So I'm not sure how many of you know about Perth in Western Australia. So here, this little golden dot on the Australian map, that's exactly where Perth is located. We are one of the top 10 healthiest cities in the world. Very well positioned in terms of, you know, the audience I'm connecting to today. It's only 2.5 hours of time difference with India. And it's also considered as one of Australia's one of the most affordable capital cities, and one of the largest number of ASX listed companies have headquarters here in Perth. So I hope that you guys have heard about Perth before. And also there's some special benefits of students who are thinking to come to Western Australia to do their higher studies or undergrad, because there's a provision of regional status in this part of the world, which you can apply your permanent residency and other things. So this is where I am right now. So this building here you see, this is the Baylis building, and this is the building where I'm sitting. This is our Crawley campus. This is the Perth city, and this is the beautiful Swan River around which the whole Perth is based around. We are ranked #1 in Australia and 17th in the world for agricultural sciences. And that was one of the reasons that I came here to do my PhD studies in 2007. We have one of the biggest in-city parks, which is Kings Park, with enormous amounts of biodiversity out there. And there's also a huge health campus, which is located on the Crawley campus as well. And we have on-campus hotels and everything, if you're thinking. And there are a lot of scholarships which are being offered at UWA. So if you are considering, then I would suggest that you please go and visit the uwa.edu.au Study Global Excellence Scholarship website, and you should be able to find further information in there. Before I get started, I would like to acknowledge that this campus from where I'm talking to you and where we do all this wonderful work, which I will be sharing with you guys today, is situated on Noongar land, and the Noongar people remain the spiritual and cultural custodians of their land and continue to practice their values, languages, beliefs, and knowledge. So I'm sure if you're studying agriculture, or you excited about biotechnology and the scope of agriculture, one of the key reasons which the entire world is thinking right now is going from population, what we have right now, to a 9 billion or 10 billion estimated to be in 2050. So there is a population challenge going on apart from all the other challenges which it has created, like the climate challenge, the challenge of limited resources, and it can keep going. And right now, the biggest challenge which we are facing, the COVID challenge. So how are we going to deal with this, and where are we headed, and how different technologies, or specifically biotechnology, is going to help us in getting there or having a better and sustainable future? So as we know, future demand food is going to increase by 70% in 2050 as the population rises, as per the World Resources Institute. So how are we going to put that food on the plate, especially when there is a scenario like Norman E. Borlaug very well said that when there is a resource restriction, you cannot build a peaceful world on empty stomachs and human misery. And that is very, very correct. And we are seeing that those challenges very, very closely right now during the pandemic, which we are facing, that how the economy is crashing, how the things, because of the lockdown, it's kind of, we're living that what we were hoping to be in 2050. It's kind of like come to us a little bit earlier. So I think it's time that we all get together and reflect on how things are going to be, and where things are leading to. So I'm a firm believer, as you can see, that a lot of my work is on biodiversity and natural germplasm resources, using what nature has provided us. And nature, while there are a lot of problems which we are facing, a lot of them are man-made, I would say, or human created problems, because we've been a little selfish, I would say. I mean, we've occupied 96% of this planet, and we're just one species out of the billions of species who also co-inhabit this planet with us. So is that the right thing to do? We haven't left, even out of the 4% we've left for the other species, we're trying to domesticate the 3% of that amount as well. We're leaving just 1% for the wild. And when we're leaving such limited resource for the other species to survive and thrive, we're breaking the food chain, we're breaking the entire system in there. And that is causing troubles. And we are in one right now. We are in a pandemic. And without going too much into the details of that, I would just my key message today is that nature has solutions. We just gotta go out there and look for those solutions and really listen closely that what should be the way forward in the future. So as we talk about solutions, we have about 17 countries which has got the biological hotspot, biodiversity hotspots, and I'm very, very fortunate to be sitting in one of that global hotspot, which is actually the world's first identified biodiversity hotspots of Southwest Australia. And how we're using that to our advantage here will be reflected in the case studies which I will be showing you guys. And that was actually the key reason for me to come to Western Australia to do my PhD, because I think that when nature has provided us solutions, we really need to go and explore carefully those solutions. And using non-model species has been my passion, because I think we got to go find the solutions or chase the problem where the problem is, rather than trying to do research with lab rats, or in plant world, we do all that research with the Arabidopsis. I'm not saying it's not useful. It's very, very useful. It's a good starting point. But having said that, there's a technological revolution which is actually enabling us to be able to even go further down the track. So why not? So as I said, Western Australia, it has beauty, which is very rich and rare, and Southwest region has the diversity at its greatest. We've got amazing, amazing native plants, which found some really fascinating ways, fascinating root systems to sort of drive nutrients from the soil, which are one of the world's poorest, nutrient poorest soils. So all these things are very fascinating to study and also explore. But what has happened is, like, being a molecular biologist, I think that whatever you see in terms of a phenotype, whatever is visual to our eyes, has always got a mechanism or a molecular mechanism underneath that. And I really get fascinated, and I'm very passionate to find that, because once you know those mechanisms, you actually get to think about the solutions or to chase those solutions in a much more fast way. And that's where the genomic resources come into play. But unfortunately, there has been, I mean, there is an enormous amount of biodiversity, but we have been just studying a couple of model systems all these years because of the limitations in having those genomic resources. And when I talk about genomic resources, and the first thing that comes to my mind is actually decoding that entire species or that non-model system at a DNA level. And to be honest with you, the current revolution in next generation sequencing, like coming from $2.7 billion genome, which was built in 2010 for humans, to nowadays when people are doing like the $100 human genomes, people are doing personalized medicine. So we're doing that for our species, we're doing that for ourselves. But what about the other things, other problems, and finding solutions for that? And that's what we will be exploring today, that how the reach of the genetics can be extended to feed the world by chasing these non-model systems. And my postdoc was in subterranean clover, which is, you must have heard about the clovers, or I used to play this game when we were little. Chasing the 4-leaf clovers, because normally clovers are 3-leaf, and so if it's a 4-leaf clover, like you kind of get lucky that day or something like that. I'm not sure how many of you played that game, but yeah, those are the clovers I'm talking about, and that will be the case study I would like to present to you guys today. So just to introduce you guys to subterranean clover, it is one of the key pasture. It is actually not one of the, it is the key pasture legume species in Australia, which is sown and is sown over an area of 29 million hectares. It's being a legume, legumes can fix atmospheric nitrogen. They're very, very good for the soil health, but at the same time, they're very good for the animal health, being very high quality forage. They've got very, very good source of proteins in them. And apart from that, they're able to grow in a diverse rainfall environment, soil types, and farming systems. So farmers absolutely love them because they're very, very easy to grow, and they're very flexible. They're not very picky about this type of soil, this type of environment, this kind of system I need, So, but apart from that, it's also a very ideal reference species for pastures or legumes for doing comparative genomics, because it's a diploid system. So it's like only got 2 copies of every genome, just like humans does. It's a small genome, so which is made up of 556 million bases. I know million bases, but still, it's if you compare it with the human genome, it's only 1/6 the size of the human genome. So the normal human genome is about 3.2 billion pieces or base pairs used if you want to compare the size of the data. And it's predominantly inbreeding. So inbreeding means that it crosses with itself. So it's when the species are crossbreeding or crossing with other species. So every time you cross with other species, you bring in the genome of that. So it becomes really, really complicated. So this one is an easy system. It's readily hybridized, and it has got a range of qualitative and quantitative traits which we could study and identify and match and go from genome to phenome, phenome to genome, etc. And having said that, although we have the biodiversity in the world, but also we were very lucky, or I was very lucky to do my postdoc in this one, because we've got the world collection. So University of Western Australia and Department of Agriculture has been collecting these clovers from all over the world. And we've got about 10,000 different accessions which have been collected from the Mediterranean basis, which is kind of the naturalized places where this clover has been growing. So we've narrowed that down to a 97 accessions or 97 types, which represents about 80% of the genetic diversity of the species, which is great because every time you're looking for something, you don't need to go and screen 10,000 accessions. You can just screen the 97, and which has kind of all the biodiversity or the diversity of that species represented in there. So that's called a core collection, and core collections are really, really useful resources which has been developed for many different species these days. And then we also added some elite Australian cultivars, about 28, because they've been doing really well in Australia. So it was, we wanted to capture the entire sort of panel. So we call this a diversity panel for developing our pangenome. So when I started working, I went and looked at many different sequencing methods, which involve long reads, short reads, or medium-sized reads, and then combining them together to kind of like read the entire genome in detail. But there's lots of challenges to get genomes together. It's not that easy to read all those 550 million bases correctly throughout. And a lot of, even after going through like many iterations, we were still sitting at a reference genome, which was published in 2017, which still had gaps and inversions and many different problems, because of which I was not able to connect the dots perfectly. So normally, like, we did capture 95% of the genome, and it's called a gold standard reference, but I was really chasing like a perfect, and I just wanted to have it like have the whole thing in one place. How can we answer some complex traits or complex questions if you don't have the full information? So that is when I met, sorry. We can move. So what I mean from pieces is, like, normally when we read all those technologies which I'm showing you, we end up getting lots and lots and lots of those reads, and we call them CONTIGS or short CONTIGS in our language in bioinformatics. But what we're chasing for is like clear chromosomes, so that you can say, okay, I found this particular, these plants have got really long root hairs, and this is the chromosome where it actually, the gene for that long root hairs is situated, and this is how it is interacting with the rest of the genome and things like that. But we're only able to do that if we got pieces like this. If we got millions of pieces like this, yes, it is useful, but maybe not that useful. So what happens is, when we do that, we try to order and orient that by using linking data, by different technologies, as I said, long reads, short reads, but that becomes a very, very expensive affair, and it's different technologies, different kind of expertise. Similar way the human genome was done, which was about $2.7 billion, the whole world united to get that together, but we can't spend $2.7 billion on every species on the planet, right? So what do we do? So there is this team at Baylor College of Medicine at the Center for Genome Architecture, Professor Erez Aidan Lieberman. He invented Hi-C. Hi-C is a really, really cool way of generating linking information. And that, I got really excited when I listened to his talk in 2015, and I had an opportunity to go to his lab and use that technology on the clovers, on my little clover plants. And we had an amazing time, but having said that, also a very, very productive clover genome assembly in 3 dimensions. So just to take you guys very briefly through the Hi-C protocol, what Hi-C does is it gives you the genome in a 3 dimension. And originally it was invented to understand the genome architecture or the folding patterns, because I don't know if how many of you know, on an average, the nucleus size is about 6 microns in humans, okay? And it's fitting in 3 billion base pairs in just that 6 micron nucleus. How it is doing that? And For doing that, it has to actually fold in certain ways so that the information is transcribed and translated through all the mechanisms which we know. And it's very specific, and that's how we kind of function and look, and our phenotypes are visible. So what HiC does is you take a snapshot of that particular contact or those contacts which the loci are making in that 6 micronucleus. And once you know that, you kind of take a snapshot of that using a process called cross-linking DNA, which we use about 2% formaldehyde to do that. And then you cut it with restriction enzymes, which are the biological pair of scissors used. Then you kind of stick that together using biotin, you ligate them, and then you just pull it through those contact matrices and then sequence them using paired end sequencing on high throughput sequencing platforms like Illumina is one of the very commonly used platform. So, and then how you analyze that data is inspired by Facebook contact mapping. So I'm sure a lot of you use Facebook and post your photos. Sometimes Facebook even comes back to you, not just with your memories these days, but also kind of suggests relationships with the person you've posted maximum number of photos. And that's called orthogonal approach and contact mapping. And that's exactly what we use to map genomes, or when I would say to build a Facebook of genomes. Okay, so I'm going to give you an example of Homer Simpson's Facebook page. Let's go through all his photos and see if we can do some orthogonal mapping using his Facebook page. So what are you going to see these days? Selfie trends. We're going to see lots and lots of selfies there of Homer himself. Some of his photos with his wife Marge, his kids Bart, Lisa, and Maggie. Of course, you're going to see Flanders, his neighbors in there, and a lot of other contacts which he kind of hang out with or come in contact with. So all these photos, what we're going to do is we're going to build a symmetrical matrix out of that. So by symmetrical matrix, I mean, let's put all the characters who you see in the photos as x-axis and y-axis, exactly the same order. And then we're going to assign a number, the number that how many times that contact appeared in a photo together. Okay, and then we overlay that with a heat map. So the bigger the number, the darker the heat map color goes. And all of a sudden, I'm seeing this little enriched square in the middle. So what does that mean? That's let's look at what does that mean. So that has got, wow, that's got the family in there. That's interesting, isn't it? So you all of a sudden you get an enrichment square which shows the family structure of this particular contact matrix which we mapped. So basically the hypothesis here is which is being used, that the people who kind of closer to each other or kind of live together or are together are going to bump into each other more, or are going to be in contact with each other at a much higher frequency than the people they live away from. And same happens in the genomes. So if this is a chromosome and there's about many chromosomes in particular species, for example, humans have got 26 chromosomes, and then you've got the other one, and we do this. And this is a HiC map for a human genome. And these little squares where you see enriched squares are your chromosomes. So am I making sense? Like, for example, if you are a loci sitting on the same chromosome, you're going to bump into each other much often than a loci sitting on different chromosomes. So just by using that approach, you're able to filter out, or you're able to visualize or bring the chromosomes together. Bingo, problem solved. It's a long-standing challenge in the genomics to be able to pull together chromosome level assemblies. And I showed you that how in clovers I went around the world, used all the technologies, poured in a lot of money, but still was not able to get a chromosome length genome. Here we go. The first species we did was a very close relative model species for legumes, which is Medicagua truncatula. And we were able to pull together the 8 neat chromosomes from a very fragmented assembly. And same thing was when I applied that to clovers. This was my, this was our gold standard genome. Look at that. We're not able to deduce any structure here. Whereas here you got neat 8 chromosomes, which a chromosome length assembly. It's beautiful. And now I can start to sort of link other forms of data which we've been collecting on clovers with this genome assembly and trying to start answering questions. And this was really wonderful. And I would like to share with you guys, and this is the basic principle behind DNA Zoo. DNA Zoo was founded by Professor Erizet Lieberman from Baylor College of Medicine. And we've got satellite branches all over the world for that now. We've sequenced 150 plus genomes. It's less than 2 years since DNA Zoo was founded on 2nd of November 2018, and we are here August 2020, and this many number of genomes have already been released, chromosome length, all open source. So if you get a chance, please go and visit the DNA Zoo website and have a look at many different examples of using that contact matrices. Moving on. So how we use that information? So as I said, nature has solutions, and nature has all this biodiversity there for us to sort of look for different variations in it, which is very, very useful. For example, I'm just showing you one kind of analysis. We call it a phylogeographic analysis, where you can link the genetic variation to different geographic locations. So in the beginning of my talk, I told you guys that we collected many different accessions, 10,000 accessions across different countries in the Mediterranean for clovers. And then we identified some of the important genes which are related to some important traits. So now what we can do is we can use all that passport information and the genomic information, and we can start to link together. We can tell, okay, the clovers with the long leaves or the clovers with more number of leaves or better biomass or yield, actually all coming from Spain. Or no, they're coming from all over the world. So it's not linked to a particular geographic location. So you can tease out things like that, which are really, really important for breeders who are developing new cultivars, that where do they go and look for. So at the same time, we were able to do like how the evolution happened in the legume family, like, how far it is from common bean, from lotus, like how many million years ago the clovers were sort of separated from lotus, like 42.7 million years ago. So you can do many different kind of studies and important understanding of the crops. Not only that, you can also do like when you identify a unique source of genes in one crop, you can actually translate it to others. So here you can see a circus plot where I have got Medicago, which is a model species, very well studied for legumes, on your right, and on your left, I have got my Trifolium subterranean 8 chromosomes, and I've identified many interesting, important traits which are important for Australia. And now what we can do is we can start to sort of look at the synteny of those chromosomes, and you can say, this part of this chromosome actually is chromosome 2 of Medicago truncatula, and that's where I should be looking for this particular trait which we are interested in. So we were able to do that for a number of genes, for a number of traits, like for example, phosphorus efficiency, which is one of the key drivers in Western Australia, being one of the poorest phosphorus soils in the world. So this whole information started a very, very exciting era for us in the clover world. We were able to do a lot of forward genetics where we knew the phenotype and we could link it to the genotype and then study the mechanisms. Just to give you an example, this was one of the key questions which I was tasked in my postdoctoral fellowship. There's a lot of loss in the environment in Australia that happens as methane from the livestock industry. A cow releases about 200 to 300 liters of methane per day into the environment. Instead of converting that food into the value, which is wool, milk, and meat, like, across the livestock, it actually goes to the environment as methane. And it has a big footprint in Australia. So we were trying to see that what are the different reasons that happens. And there's many different aspects that have been looked for that, to resolve that population. And one of the biggest one is diet. And as I said, clovers are one of the key pasture legume species, which the livestock depends on. So what we did is I phenotyped that trait. I did a lot of in vitro fermentation assays and then looked at the QTLs, which were responsible quantitative trait loci, which had some genes, which gave us links to understand the mechanisms that was happening. And we were able to identify many single nucleotide polymorphism. And one of them was this one based in the haplotype block, which is the region where the crossing over doesn't really affect much. And that was a very stable marker for us to develop, a biomarker for us to develop for breeders to look for low methanogenic traits or high methanogenic traits. So that is a very, very good way to sort of do animal and environment friendly breeding. So now we don't need to fistulate the sheep in cows to be able to study that and do the IVF TSs. We've got a simple marker which we can use, do the PCRs, and everybody's happy. We can do reverse genetics. For example, if you do know the genotype, you can actually go to the phenotype and test that whether that genotype in other species will also give you the same phenotype in the species which you're interested in, and then the mechanism, and if that is going to be the same or different. So for example, in Arabidopsis, there was Oxford University find that if you turn on the master regulator RSL4 transcription factor, you can dramatically increase the root hair length 5 times, like from 2 mm to 5 mm, 10 mm. So I thought, okay, can we do we have that master regulator in clovers? It's not that far off from Arabidopsis. And we mapped an ortholog in there, which we try now. We've been doing the CRISPR genome editing to be able to sort of increase the root hair length, because we know by studying different clover varieties or different pastures that the pastures which have bigger root hair length are actually more phosphorus efficient as compared to the clovers which are less root hair lengths. So this is just one example of doing your reverse genetics. You are able to translate knowledge across different species. Similar way, we do a lot of transcriptomics, which is the study of the RNA. I've got the papers in here, so you guys please feel free to read the full article so that you can develop an understanding. But we did a lot of gene network analysis for phosphorus acquisition studies. We use hydroponic system for that. We also used electron microscopes. We were very lucky to have a very big microscopic center for microscopy here at UWA. So we are able to do very, very high resolution studies here and link them to the genomic gene expressions which we are getting. Due to time constraint, I will not go into the details, but I'll be happy to take questions, or you're very welcome to go and read the full articles here. Similarly, it's, we got very, very less time. So if we are always in a hurry, and sometimes, some crops take forever to develop one generation, but when you want to develop things faster, you need to grow them faster and get them to flower. So I was very fortunate to have people who do this, has this expertise in the same corridor I sit, and they help us develop an in vitro assisted single seed descent method by which we could take 3 to 5 generations per year. So that kind of fast progress of a work. When they were doing that, I was really keen to know that when we are doing this fast forwarding from one generation to another, that how that is linked at the molecular level or the transcriptomics level. So we also studied the RNA, and we were able to map those pathways, and we published that work as well. One of my students, master student, came to me and he said, I want to apply remote sensing in agriculture and somehow work on clovers. And I'm like, very aspirational. What do we do? And look, he came up with this idea. We bought, we tried to apply the NDVI method, which is one of the methods used by NASA for doing the vegetation calculations. And what we did was we converted, we bought a $500 camera from eBay and just changed the filters, put the NDVI filters in there. And then he took photos in the field of all the 125 diversity panel, and we were able to do very, very accurate biomass and many other measurements. So this is the clover which is growing, and these are the photos of that one which you see. And that was, and then we were able to do some correlations with many other many other data which have been collected on the clovers, because every single accession is coming from different part of the world with the passport data in terms of all the biological parameters and things. This is some really, really interesting stuff, which was also published in PLOS ONE last year, or early this year. So if you're really interested to understand how core collections are useful tool for examining trait diversity and plant breeding solutions, developing plant breeding solutions, I highly recommend this paper for you guys. And then one of my master students, she worked on the boron tolerance, and we were able to link the boron tolerance genes and identify, how they're linked in the clover genome and able to provide valuable markers like we did for methane work. One of my students has been working on insect pest resistance, where I have already identified one of the key genes that are responsible for pest resistance. And we did big trials here. And now we are trying to do some gene editing in terms of knockout to really validate that that is the gene which is doing the thing. So I mentioned CRISPR many times. I'm assuming that you guys know that how CRISPR works, and how it is really a very, very useful tool these days in the gene editing space to be able to validate your identifications. And how we use CRISPR, we kind of use it in a novel way, where we use the CRISPR to not just do the loss of function, but also the gain of function. So for example, when we design our gene constructs and we attach them to the promoter region, we get a gene activation or a gain of function. Whereas if we do the same for somewhere else positioned in the gene, we end up getting a gene inactivation or the loss of function. Integrative genomics. So this is something I really want to stress about, that, it's always a good idea to parachute out of this small little problem which you're trying to resolve in your studies, and trying to look at the bigger picture. And sometimes you can do things in a strategic way to be able to make a bigger impact. And the one key thing which you will always need as a key thing to make a bigger impact is collaboration, working together with others who got the similar sort of passions or similar sort of drivers or goals in life. And that's exactly what I did. I collaborated with a number of people across Australia and worldwide who were interested to study clovers. And we were able to, while we were mapping all the genomic side of the things, and those people were doing other different traits on them, we combined this all together and created this integrated platform for clovers. So now we've got a very powerful system which is all connected, not only the genome-wide association studies, we are able to do diversity analysis, we're able to do fingerprinting, hybrid testing, we have a stable transformation protocol for doing genome editing, we can do rapid generations, and then by doing all this, we are able to transfer a lot of knowledge from someone called clovers who nobody knew before, and now it's become kind of the central point for a lot of research worldwide to be translated across to other clovers or other related species in the legume clade. So that's when things become really, really powerful, when you network, when you join with other people, when you show interest in their work, and then think about ways how you can connect the dots. So this is what I see as the future of agriculture, with all the technological advancements. We don't probably need to do a lot of phenotyping upfront. We will be able to develop in silico models, genome scale models, where we will be able to, look at all different kind of parents you could breed with, and look at all sorts of fermentation combinations of hybrid models, and do phenotypic analysis in silico before you actually go to the field. This will cut short a lot of resources and time, and I think has got a great potential in the future. So I think I've got about another 10, 15 minutes to wrap up. And I'll very quickly go beyond clovers, that how we are using the similar sort of approaches in my lab and applying the same principles, but things beyond clover. So we've been working on a lot of iron fortification genome editing scenarios, specifically in lentils in collaboration with one of my friends in University of Melbourne. We've been looking at waterlogging tolerance in peas, so that in Bangladesh, Shaheen, he was a PhD student from Bangladesh, so that they can take more number of crops in a year. So when the rice is harvested and they want to go take a next crop, but there's water in the field, so how can you develop varieties of peas which are, you know, waterlogging tolerant, so that you can just straight away sow them in despite of having a lot of water in the fields, and they still grow. So things like that are very, very useful and kind of things sought after by the plant breeders. So to use the resources to the maximum. He did some wonderful work which has been published as well, so please go and have a look at this article. Similarly, also looked at the 2,4-D resistance mechanism in wild radish, and this was a really fascinating work with one of my colleagues here, Danica, where we did a lot of transcriptomics and was fascinated at how auxin signaling and plant defense are connected. So if you're interested, please go and read this article. You will be able to know a lot more about this in that. Right now, I've got a student who's working for a DNA-free genome editing system for wheat, so that for Australian wheat varieties, and we're chasing better yield and better phosphorus efficiency. Again, just another example beyond clovers. Not just there. This is really interesting. I happened to have a coffee with one of my colleagues where we were he's a clinician and looked at the wound healing process and why sometimes you get scars and sometimes you do not get scars. So as you know, you can't go for setting up an experiment like this, you cannot go and scar people, okay? But you can do that with plants very, very easily. So what I did is I just out of interest looked at, I picked up soybean genome because that's very well studied genome, and trying to look at the orthologs genes that map across the soybean gene and the human genome. And I found there was about like a 10% overlap in the two, which was really interesting. And we started to see that if we can wound plants in different ways, shallow, short, long wounds, then can we study that mechanism? And that was a very fascinating study. Hopefully we will be able to publish that soon and you'll be able to read that in detail. And very quickly, this is all the work which I've been showing you, which have been taking us all these amazing mechanistic insights into different processes, have been using a very traditional transcriptomics method, which we call as bulk RNA sequencing. But recently there has been a real amazing innovation, which is called a single cell genomics, which means that you can actually study every single cell and every single gene that expresses within a single cell. How cool is that? So that basically means that you can go at a very, very high resolution, and a human body has about 37 trillion cells, and all of the cells, although they have exactly the same genome or the 3 billion base pair letters that write the blueprint of that genome or the cell, but not every cell behaves the same. So for example, every cell in your body doesn't become a heart cell or a kidney cell. They all have different functions and they develop different organs. And how does that happen? That is what you can study using single cell genomics. And that's why it is called as one of the paradigm shifts of this century, because it is able to tell us so much more. So we've started a wheat cell atlas project where we are actually looking to develop a full reference single cell genome map for wheat. I strongly believe that single cell genomics hold the potential to revolutionize the way we characterize complex cell assemblies and study, you know, how the pathways, how the functions and the crosstalks happen within those complex systems. So watch the space. But having said that, as I just mentioned to you guys, 37 trillion cells in a human. We don't even know how many of those, how many different cell types in wheat and in any other organism. We are still mapping the human cell atlas. So there's like with this huge amount of data coming together, there's like a lot of analysis tools which are becoming available, but but it has been a real headache when you start to develop this for a genome size like wheat. Not sure how many of you know, but wheat genome is about 6 times the size of the human genome. So it's about 17 GB compared to the 3 GB human genome. And it's a hexaploid species, 6 copies of every single gene in every wheat cell. So it's quite complicated. And we had to go on a journey to sort of, we generated the data, but how to analyze the data. None of the tools which were working for human genome and cell atlas analysis were working fine with our. And so we had to go and we have written our own package. So that's where the soft skills come in very, very handy. We work with the computer scientists very, very closely. And we used, because it was massive amounts of data, we also used some of the machine learning for self-organization of those and doing the dimension reduction for the single cell data. So that's something also we hope to publish very soon. And the last bit of my talk where I want to say that, you know, it's amazing to be able to explore the nature and what nature has given us. And synthetic biology is an emerging field which has got a great potential, especially microbial cell factories. They've got very high value applications in terms of the biopharmaceuticals and drug discovery industry. So if you've got interest in the microbial world, I think this is a perfect time to get started in that area, and combine that with the synthetic biology advances which we are doing in our lab. We're trying to do things ex planta using, you know, yeast, algae, and many other microbial systems where you can do productions and also kind of scale them up really, really fast. So basically, for doing all that and for handling a lot of data, you need a multimodal and a multi-scale approach. And I call it data for life. And it's innovating life from cell to shell. So basically, you're studying cells, but you need a lot of information and a lot of processing in the shell to be able to do that. So you need to go and switch across micro and macro levels really, really readily and fast. For that, you do require a lot of network architecture, infrastructures in terms of high performance computing, AI, machine learning, all these things these days. And they go across different disciplines really, really well. And I would say that these are the areas for students to sort of develop further skill sets into while having an interest in biological sciences, so that we can move things forward and be ready for 2050, right? So all this is causing a data deluge, and we're calling it an informatics bottleneck because the data is coming from all different directions, and we are sitting in a very classical 4V big data problems. So there's volume of data, which is the first problem. As I said, we don't know how many trillion cells in a wheat genome, which is 6 times the size of a human genome. And you know how many different varieties of wheat we might need to study to find the one which is perfectly suitable for this particular climate or this particular soil type and things like that. There's a variety of data, like as I said, long reads, short reads, this reads, that reads. Velocity data is generated at all different speeds and transferred at all different speeds across the globe, and also the veracity or the trustworthiness of data, because you know when data is transferred from one platform to another, there's always a loss, or there's some sort of file corruption happens. So we've got all these classical challenges. But having said that, in Western Australia, we're very, very fortunate to have a POSI Supercomputing Center, which powered us with amazing supercomputing resources like Magnus. We've got Zeus, which is amazing for visualization side of the thing, especially for 3D genomics. We've also got cloud, so we are able to work with people across the globe by giving them access to our research cloud and collaboratively work and learn with people. That has come very, very handy for GNA Zoo, which is a global project, and also for data storage. It's one of the most expensive things in the world right now is to store your data, so we are very fortunate that UWA is a partner organization to POSI Supercomputing Center, so we get access to all these amazing resources. And not only that, we believe in reproducible research, because if I'm generating a data, which I'm analyzing here on my cluster, and if I give that data to you, you should be able to analyze the data and get exactly the same results. It's very, very important to have reproducible research. And for that, we practice containerized bioinformatics so that it's independent of the compute environment, and it's kind of like readily shareable and transferable to everybody else we work with. With that, I would like to acknowledge all these amazing people and organizations who've given me an opportunity to work with them, came along on my journey, on my passions, and made this work possible. And also to my son, here he is working with Abdi on the remote sensing side of the things. When he was only 1 year old, he's always been there, and all my colleagues who've been very, very accommodating for a young family, which I have to care for. So with that, I would like to say thank you to all of you for listening, being so patient. And this is how we do agriculture or other sciences at UWA. We try to enhance genetic gains and understanding through innovations and also paying attention to the nature around us. Please feel free to follow me on Twitter at dr_Parwinder. I'll be very happy to take any questions you've got at this moment. Thank you.
Thank you, ma'am. It's always when I view your talk or listen to your talk, it's feel like we know very small. it's many things we can do, and is a great opportunity, because I know your PhD and postdoc work. It's create a field, like it's a long work and is a very systematically way you did that phase of your research life. So it's create very much and very interesting new field. So ma'am, there is a few questions we have selected from our YouTube channel. I going to paste it into the our chat box, in Zoom chat box. There is a I just wanted to paste here for you. You can choose. There is a mentioning that there is an arrow, there is brackets. Brackets mean the name of the attendees and the questions followed by the question.
Mm-hmm.
Can you hear me?
Yes, I can see the chat box now.
Okay.
So so I've got a question here which says, can you share how you sample from 10K accessions to get the core collection? What are the parameters? And the question is from Alvarino. So I hope I said your name right. So, but so this is a method called core collection development, and there are a lot of publications around that, how you develop core collections. But the key is that you look at many different layers of data collection to do that. For example, for our core collection, particularly, we had 3 layers of data. We had ecogeographical data, like where the accession is collected from. We had agromorphological data, which is like, all kind of parameters which were collected alongside when it was collected in terms of leaf, petiole length, leaf diameters, everything, agromorphological characters. And then we also had bioclim data associated with those geographical locations. So there is a website called Bioclim where you can actually put in a particular geographical location and you can get rainfall, all sorts of parameters from that, like what is the height of that place, and what is the amount of annual rainfall, what is the wettest month, what is the, all sorts of things. And then lastly, we also generated a lot of molecular data. And then using all these parameters, you run it through many different workflows, and one of them is MStrat, and then you can develop a core collection. So there's a papers published on that, so please feel free to read for further details. I hope I've answered that question and explained to you. So next question I've got from Iswar Agri. What are the benefits of contact mapping? Okay, Iswar, as I explained, I hope I explained it well with the Homer Simpson slide. So if you do know that which part comes in contact with which part more often. You do so you do know that that means that those parts probably been sitting close enough to each other, and that helps you understand, you know, the architecture of the genome. So that's what exactly contact mapping is, like how far that contact or how often they contact each other. Can we use high-C technology in plant or animal species which are not sequenced? Absolutely, that is exactly what we're doing, and there's lots of examples, hundreds of examples for you to go and explore on on the DNA Zoo website, which I shared with you guys. What makes, I've got another question from Jamir. What makes clover stand out from among the legumes? Can it be used as an important legume in India? It is used as a legume in India. I don't know the amount of area under that, but it is worldwide used, and especially in the Mediterranean environments. And it's really, it stand out to me, and I think to a lot of other scientists, because it has got many unique characteristics, like, as I said in the it's very high in protein. It's a legume, it's very good for soil health, and it has adaptability, which is across different rainfall regions, some soil types and farming systems. So that makes it very, very unique. Instead of single seed descent method, can we use speed breeding technology? Absolutely. It's just, it's used anonymously. You can call it single seed descent because it's a very scientific way of doing it, but it's actually speed breeding. You fast making it fast, right? So it's the same thing. Dr. Kanak Saxena is asking which software is used for integrative omics. It's not one software. I just made up that term because I kind of connected. It's just my own creation. There's nothing like that as integrative genomics. It's just that you connect different dots, different pieces of information which you've collected through different kind of omics technologies. I've got another question from Balkis. How can genetic engineering ensure environmental sustainability as well as increased food productions when pressures on environmental resources like land and water is growing? So to answer that question, I would like to take an example of the root hairs, which we have been, we've been trying to extend the root hairs. So what happens is if you have better root hairs, you're able to find the micronutrients, even if it is far away from you, if you're a plant, right? So that way, even if there's minute amounts of phosphorus present in the soils, you're able to find it and use it for your growth. So that's good, right? Because then you don't need to put a lot of phosphorus into the soil, which causes a lot of problems, because if you put too much micronutrients in the soil, they go in the waterways and then they cause eutrophication, so many environmental hazards. So less is more, but you've got to be able to have ways to find that less, right? And that's what we are empowering our plants with using genome editing. So nature has always selected. Nature is even now selecting among humans who can survive COVID. And, it's the same thing. You do it yourself or nature does it to you. But there is a fine balance of upsetting that system. So, yeah. I hope I answered that question. So next one is from Anila. What made you to choose to study clover over other plants? Okay, I think I had a slide on that one. So clovers, it's a diploid species, 2 copies, small genome, inbreeding. So there was no it's not changing every time, it's crossing with the other plants. So there was a lot of advantages to study clover, because when you're starting in your career and you're exploring there was a lot to learn, to be honest, and you can do what you can do in a certain amount of time. So you have to choose carefully a system, at least a system is established or a plant is good, so that you can try other things. So there's a limit to what you can learn and explore, right? So that was one of the reasons. And I absolutely loved the clover connection with the methane and environmental impact I could make by, doing some good work in that area. So that was one of the reasons. And of course, the amazing people I worked with. Can clover be helpful in agriculture area during drought time, and will it be able to fulfill food requirements? Sorry, it's a pasture. It's humans do not consume it. It's for animals. It's a fodder species. But yes, it is. It has. We have identified genes which can make it survive in drought or less water, and which could be useful if you are able to transfer those genes or translate that information into other species like wheat or which humans eat. So everything is connected. You find something here, and then you can go reverse genetics, as I showed you an example, and maybe that would be a useful thing to do in future. How AI and machine learning can be integrated in plant research. Okay, so there are many. I mean, it's really your creativity how you integrate it. How I integrated it is I'm trying to use self-organization maps, because there's so much data which is coming out of the single cell genomics, where we really need machine learning and AI to help us, because trillions of cells, and every cell we generate about 50,000 reads. So you multiply, I can't even multiply that number, like 37 trillion times 50,000 read for every single gene, and there's about 20,000 genes in every single cell. So that's kind of like a really big number. So that's why you need the computers, you need high performance computing, AI, machine learning, and all these tools. Dinesh saying, can clover genome database help to modify other crops for abiotic stress tolerance? Absolutely. So as I showed you guys in the integrated slide, that you can translate information across very easily. And that approach is called comparative genomics, and you can do that. You can map orthologs, homolog genes, and then look for them in other species. Okay, so we got Diriba. In terms of code collection, there is different factors which cause genetic drift and bottleneck in population. Absolutely, there are. And I mean, we haven't studied that extensively in the clover, that thing, but there will be more students coming hopefully on this journey and taking it over me, and go on that path as well. Huzefa Bashir is asking, how can Hi-C be used to assess the sustainability of a genome in some horticultural crops? So Huzefa, there is a lot of horticultural crops. I just recently published the genome of guava, and then we try and apply the Hi-C technology to that as well. So there's you can use, it's a technology, it's a tool, you can apply it to anything you can handle, or you specialize in. So there is no restriction of species, and the protocol is publicly available, open source. Everything I have presented is open source research. All the data is open source, and so are the tools and technologies. I hope I've answered all the questions, and but still, if you got more questions, find me on LinkedIn or Twitter, and stay in touch.
Thank you, ma'am. Now we have moved to our, we should move to our interview session. There is a short interview of you, because so many younger students are there. So it's always a motivational for them also to learn and how you build your career, and that's all. So I request Shoma to interview to you.
Thanks.
Shoma.
Hello, ma'am. We are now moving ahead to the interview section of today's webinar. Today is our 13th webinar of the series. We are lucky and honored to have with us a very distinguished scientist, Dr. Parwinder Kaur. Ma'am, you're a very successful researcher, having achieved so much at a young age. Please share your journey so far as a plant scientist.
Shoma, thank you so much for having me today, and for the kind words you've said. I think all I have to say is there is no shortcut to hard work. As you can say, it takes a lot of work, but it does also take strategy, like in terms of like where you got to have clear questions. You just can't be mucking around, and it's very easy to get lost in a lot of work and not be productive. So what I always say to my students is it's always a good idea to have a clear and clear direction, but also having very clear hypothesis that exactly, because we're doing science here. So if you do not know what you're chasing, it's not going to be very productive. And as I also said that mentors and your networks are really, really important. It is as important as doing the hard work, that you got to be connected to the right people who can show you the right path, but at the same time also mentor you. Learning from somebody's other experiences and mistakes save you from repeating the same in your life and wasting that much amount of time which you could have used to do something different or more productive. So find yourself a mentor, find someone who's got the same passion like you and can guide you on that journey. It could be a very fascinating and productive journey if you got that mentor right. So I would highly advise people to find a mentor.
Thank you. In the era of big data and artificial intelligence, how do we progress with respect to modern research and its field application?
So modern research is no more the same, like, just screening a few cultivars for one particular trait, or just go and grow some things in the field and see how they're doing. It's a lot more than that. As I said, like, probably we are moving into a future where most of the agricultural research might be just in silico. I mean, because we do not have the amount of resources to grow such big field trials and things. But whatever you do, it's not going to completely replace that. But whatever you do, basically, there will be so much amount of data that will be generated from those few field trials that could be applied across a number of ways, in many creative ways, to do much more than what we could ever think of. And that's why the power of cloud research clouds is coming, because you can share data so fast, you can transfer data so fast, you can connect across the globe with people.
Right.
Basically, you can harvest much more. So, and it's, remote sensing in agriculture, high performance computing in agriculture, able to do like things which only could happen in plants. Now we can do that. We can take that mechanism and put it in the yeast, synthetic biology.
Right.
And you can make the yeast do the thing which you had to grow thousands of plants for. So
Right.
I mean, all these things, this is I think this is where the future is taking us, and it's very exciting.
Definitely. According to you, which sectors of plant science research and agriculture will prosper in the near future?
I think it's a very hard question and very hard to answer in a few words. But I would, as I said, I think it's data, it's coding, it's probably not going to be the simple degrees like which we used to do. Like I do a degree in plant biotechnology, or I just do a degree in plant biology. It's got to be combined things like computer sciences and plant biotechnology, or agricultural technology, business while you do technology development or biotechnology. So we've been thinking about all these things, that what exact things we should be offering in terms of skills to our students, the next generation students. And I'm very glad to announce that this semester UWA has announced a number of double degrees which students can explore, like combining, like for example, masters biotechnology, which I teach. It's a combination of a unit where we offer the business and biotechnology together, because we see these biotechnologies as the budding entrepreneurs. So they got to have a little bit of a knowledge of how to develop or set up a new business or a startup. So all these things, you got to learn while you study. So these are the skills I would highly recommend students to look for. Yeah.
So interesting. Ma'am, we would all love to know how you build your career as a scientist. We would love to know more about you.
How I built my career. Look, I've actually let the life happen to me, and it has taken me places. I studied agriculture from Punjab Agricultural University. I was a 2000 batch. 2004, I completed my bachelor's in agriculture with an honors degree. I specialized in plant protection because I was always fascinated that, how the plants are able to handle all the kind of insects, the weather, and diseases that all come to them. They're out there facing everything, and how do they do that? So that was kind of like my curiosity, which took me to that degree. After that, you have to pick up a specialization. So I picked up entomology, studied insects because insect pest specifically. Um, University of Western Australia had PhD scholarships, which I applied for, and I came here to study plant pathology. And after that, I even went to Telethon Kids Institute and like worked in the immunological area. So I think what I haven't been very strategic, but I think the way life happened to me, it has been very strategic, and the continuous thread, which I would say has been running across my passions, has been DNA. And I think that's an interesting thread that also connects all different forms of life that exist on the planet. So that's exactly how my career's been. It has taken me places from pretty much every life form on the planet, and I'm very, very, very grateful for that journey that happened to me. I really did not choose it or did not plan it.
So beautiful to hear from you, and I'm definitely excited. And after watching your presentation, first of all, I was very excited to visit Perth. It's so beautiful.
Please, yeah,
like
we love to have visitors here. Yes, it's beautiful weather here as well. So yeah. And if you're passionate about agriculture, definitely look at this background. This is wheat fields in WA. We are the golden wheat belt.
Oh wow, beautiful.
Thank you.
Okay, and we hope to hear from you again soon in the future if you have new scientific discoveries, and we will love to hear from you again. Thank you so much for being here today. Let's go back to Shubha now. Thank you.
Thank you, Shoma. Thank you so much. Thank you, everybody.
Thank you, ma'am. It's a very positive feedbacks are there. I can't copy paste all of it, but I collected few feedback for your talks, and everybody
It would be lovely to be able to see that what people think, and if they've got any suggestions, I'm very open to any feedback. And because it's always a learning experience, and you guys have got a lot of young minds connected here who've been listening to me. I wish if I could literally talk to all of you, but yeah, it's been a pleasure.
And we, as you said, that collaboration is a very important. So it is. You can see that it is a collaborative work. Like Jacinta and Liberty, we are doing a training in Erie, Philippines. So since we are friends, and now we are connected to, online. Neha Shoma is my MSc friend. So we are. Somehow we are connected and build this platform, and we all are doing our PhDs in plants. So we are plant loving.
Please come and visit us. We've got some fascinating plants here for you guys to explore as well in WA. So I hope I have convinced Shoma to do a PhD now. I personally I feel that collaborative work is more important, and that is the reason we now we are planning to give some ideas through the soft skill development, which are very important, and we learned through the struggle we had made. We learned how to paper write in very difficult way, but we can learn. So we need to share this information to everyone, to younger generation, to school, to college, and to JRF and SRF. That's Absolutely. Why we also included soft skill, because it's not related to plant biology, but without soft skill, how we can improve our plant biology in writing, poster, and that's why. So I think it's not related. Actually, we are promote plant science, but I convinced my team that we should go for that, because Jacinta and Liberty may have the idea, but
we need to need that type of skill set, like collaborative work and open-minded. That the nature. So
Absolutely. As my 7-year-old son tells me that, Mama, sharing is caring. And we really need to keep reminding ourselves of that thing. Sharing is caring, and science is all about this. So it's we got to share what we do, and we got to connect with the like-minded people. So thank you so much.
Thank you, ma'am. It's the end of our webinar. It's we clip your interview section differently and your talk and upload it in our website by tonight or tomorrow, and you can see the post webinar feedbacks and updates, and very much I feel that it's a very new in this platform, but many people are connected. We have about 4,000 plus subscriber in YouTube in 1 month. I think it's within 1 month, and about 3.5 thousand followers in Facebook page. It's within 2 months, I think. So I think we are in good direction to promoting plant science, and all of you, all the speakers are very I personally and from my team are thankful. So I request all my teammates say bye to our speaker, Neha, Jacinta, and Shoma. Liberty is out of meeting because it's a late night and she has some work to do.
I gotta go pick up my son as well. Wait for the school pickup.
Okay, ma'am. Bye. Bye.
Thank you.
Bye. Now I'm thanks Neha, thanks Jacinta, thanks Liberty. Now we are going to a short instruction round because there is a soft skill development training of the soft skill process of their webinar is there. So I'm request Shoma to elaborate some of these facilities. Thanks, Neha.
Hi everyone.
Thank you, Shubha.
We have some very exciting news for all the college students and PhD scholars. Many of you have been writing to us for quite some time. Okay. Okay, my mic was off.
Shoma, your mic was off and I can't listen anything. So please, can you please repeat?
Sure. I'm so sorry. I'll just begin from. Hi everyone. We have some very exciting news for all the college students and PhD scholars. Many of you have been writing to us for quite some time for guidance related to soft skills in academia. We start the college level naively with the vision to achieve our academic goals for ourselves and the society. Little do we understand that the path has many ups and downs, both at the professional and personal level. For example, we lose time looking for the right direction, resources, and strategies to succeed. Bioingene is bringing a webinar and panel discussion where we will discuss the importance of soft skills and how they help us to become better professionally and personally. The panelists will talk about the challenges they faced in their master's and early PhD lives in coping with the preparations to become professionals in a new work environment. They will also discuss how they overcame it. The title of the webinar and panel discussion is The Importance of Soft Skills in Academia. The webinar will be presented by Dr. Konkona Ghoshal. Dr. Ghoshal is a researcher, educator, and a former lecturer at UCLA, that is the University of California, Los Angeles, USA. As a lecturer at the UCLA, she taught a course called Introduction to Collaborative Learning Theory and Practice. She has expertise in teaching soft skills such as skills and strategies related to collaboration, communication, group dynamics, and team building, learners' mindset, students' motivation, and mental well being. Soft skills help us to grow systematically and become lifelong learners. It helps us to design our life on the lab bench and beyond. I would personally request all the students to attend this webinar as it would help you to be aware of the necessity of soft skills so that you can holistically enjoy your studies and research. I hope it will be an eye-opening experience. You can register for this webinar from our Bioingene website, and registration links are also available on our Facebook page. The webinar is on 5th September 2020 at 10 a.m. Indian Standard Time, Saturday morning. Hope you enjoy the webinar. Thank you. Let's go back to Shubha now.
Thank you, Shoma. Now I screen share today's password and also the upcoming webinar. There is a lot of I hope everyone get the password, and those are coming at late at the end of the webinar. I'm wanted to request all of you again, please come at the beginning, because it's not only to collect the password. You should learn something from the webinar also. So there is a feedback link and password. Password is a 31419. For those are emailed me after the webinar that she or he could not get the password. So this display there, I displayed this password. Our next webinar on 17 August 2020 at 11 a.m. Indian Standard Time on microprotein. Speaker will host our present from the webinar from the Belgium. There is a ongoing others webinar registration are ongoing from our website. Go to our website and enroll whatever your interest and what is your upcoming webinar. You can see everything in our website. It's bioingene.com, but it's yet to be developed. So there is a link provided to our alternative websites. And those are the register. I already mailed every information to your email. Today's password is 31419. And we introduce one other webinar and panel discussion on the soft skill. Here I intentionally put the panel discussion because you have to know the struggle. You have to know first the struggle, then you can only understand the importance of soft skill. It's a very common process in first world countries. Now they have started it from the school level. Obviously, graduate and undergraduate students know the soft skill. From the bioengineering team also, I know that Jacinta, Liberty, they are habituated about the soft skill. So it's basically we encounter with that problem every day in our lab, in our life, in our school, colleges. Soft skill will help us to understand the problem, to identify that this is a problem and we have to struggle, we have to cure it, or we have to try to address it. So for easy examples, is beyond the writing process, is beyond how to make the poster, how to make the presentation. These are the soft skills, obviously, but there is more in soft skill, like how to manage your stress, how to manage your anger, how to collaborate with others, how to present your paper with different countries, or collaboratively write your paper more accurately. So there is a panel discussion, and all this panel is enriched by all PhDs and postdocs and scientists. So we have 6 names in that panel discussion. We try to more names and more speakers for the participation in that panel discussion. First, we discuss about the struggle and importance of soft skill. Then Dr. Kankana Ghoshal will deliver her talk. She is an expert on soft skill education system in Los Angeles University of California. So it's a nice opportunity to us, and I'm very happy that we can connect Dr. Kankana Ghoshal for these purposes. And I request every students here, scholars, JRF, SRF, PhD students, BSc, MSc, please share this information to your friends also, because it's basically it's a free webinar and open to all, and there is hosted in YouTube, so there is no limit of attendees. So I highly recommend, please share this information to your colleague, to your friend, to your juniors, seniors, to understand that how soft skill is important. And I know that most of the developing countries has no dedicated course for soft skill development. Those are the very few and very limited. So it's a nice opportunity to us to learn the area of soft skill and importance in academia. We all know that it's a very important, but now we able to understand that dimension of soft skill, because I also not know how many soft skill are there. I know few, and I try to know more. That's why it's in my intention, and this is the first time I included something other than plant science. But I'm assure you, all the plant science scientists, all the plant researchers are need this soft skill, and those have this soft skill are flourished and do their research very positive way and constructive ways. So we request all of you, please come on that 5th September 2020, and this time is 1 hour ahead. So those are coming at 2:30 and late, that day it's very difficult for you guys. Please come at morning, because that time password link also be closed. So come at 10 a.m. learn soft skill. This registration is ongoing in our website. This flyer will available tonight in our Facebook page, in our LinkedIn, actually in my LinkedIn account. We don't have yet LinkedIn for bioingene.com, but we very soon we try to create LinkedIn and other social media for the Bioingene. So I know that those are coming at 2:30 and above. All are given. Those are asking for that. I'm not giving the feedback link, or I'm not giving the password. It's all are giving in the chat box. Please collect from the chat box, call and find in the old one. And this link also available in our website. If you know the password, go to the website. This link is available on just beneath the talk. This link actually active for few more times. because it's a self-destructive link. Within 2 hours, this link will deactivate it. And it's very necessary to control this type of phenomena and ensure your honesty. Bioingene not your certificate manufacturing software, or not not something you can just come at the end and collect your certificate. So these are the password and link. Thank you, everyone. I'm going to close this meeting and collect the link from our website. 31419 is the password. See you on 17 August, webinar on microproteins. Thanks, everybody. This link is not going to be shared anymore. And very soon, YouTube broadcast also be closed. So link is available. I again repeat, link is available on our website. And password is 31419 for today's webinar.